Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Wielding the Double-Edged Sword of Cardiac Biomarkers in Clinical Trials. My name is Ryan Muse, and I'll be your X-Talks host for today. And today we have a discussion with Dr. James Januzzi, MD. And today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. And the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using this questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, please contact me at any time by sending a message using this same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Medpace, who developed the content for this presentation. Medpace is a scientifically driven, global, full-service clinical contract research organization, CRO, providing phase 1 to 4 clinical development services for the biotechnology, pharmaceutical, and medical device industries. Medpace's mission is to accelerate the global development of safe and effective medical therapies through its high science and disciplined operating approach that leverages regulatory and therapeutic expertise across all major areas. And now, without further ado, I'd like to hand the mic over to our first speaker today, Richard Lee, MD, Senior Medical Director, Medical Department. You may begin when you're ready.
Thank you, Ryan, for that kind introduction. And on behalf of Medpace and my colleagues, I would like to welcome everyone to today's webinar, which I think will prove to be an excellent discussion on a very relevant topic in cardiovascular clinical trials, specifically cardiac biomarkers. And we are delighted to have this unique opportunity to engage our audience. I would like to thank our speakers, our audience, for their attendance. I am Richard Lee, Senior Medical Director at Medpace and boarded in cardiothoracic surgery. Co-presenting with me are my colleagues Kelly Milham, Director in Clinical Trial Management, and Liz Moore, Cardiovascular Advanced Nurse Practitioner. We are also very honored and delighted to feature Dr. James Januzzi, Staff Cardiologist at Massachusetts General Hospital. He certainly is a luminary in the field of cardiac biomarkers. He is the Hutter Family Professor of Medicine at Harvard Medical School. He serves on the Senior Cardiometabolic Faculty at the Baim Institute for Clinical Research. Dr. Januzzi has also led numerous steering committees, clinical endpoint committees, and DMCs for heart failure trials. So thank you for joining us today, Dr. Januzzi. Today's agenda is outlined. To start our presentation, I will provide a brief overview of the rationale for cardiac biomarkers in clinical trials. Dr. Januzzi will discuss strategies to gain efficiencies in clinical trials using biomarkers. Liz Moore and Kelly Milham will present lessons learned from heart failure trials using biomarkers and mitigation strategies. And we hope to have time for a few Q&A at the end. So the aim of cardiac biomarkers has traditionally been focused on the use as a diagnostic tool. Cardiology has been limited to essentially 2 biomarkers, troponins and natriuretic peptides. For example, cardiac troponin is used for the diagnosis of MI and myocardial cell injury. Natriuretic peptides like NT-proBNP or BNP are used routinely for acute and chronic heart failure. Moreover, the literature supports use of cardiac biomarkers as prognostic indicators for major adverse cardiac events like non-fatal MI, heart failure, and CV hospitalization, and all-cause and CV mortality. In clinical trials, most commonly, cardiac biomarkers are included in efficacy and safety endpoints. For efficacy, biomarkers identify a patient's quantitative response to study drug. For safety endpoints, cardiac troponin, either high sensitivity or standard, are used as markers for safety and myocardial injury and toxicity. Increasingly common is the application of cardiac biomarkers to determine trial eligibility, to establish common quantitative baseline characteristics. For heart failure trials, natriuretic peptide cutoffs are now routinely used. And as I mentioned, cardiac troponin is commonly used for safety, but also now being used for heart failure subphenotypes. And then finally, cardiac biomarkers may facilitate matching the study drug mechanism of action to the biological effect using a so-called precision cardiology approach, which we will hear more about by Dr. Januzzi. So Medpace has a rich history of conducting CV clinical trials, and based on our extensive experience, we wanted to highlight both sides of that double-edged sword of biomarkers. So biomarkers, cardiac biomarkers, can be your friend as listed on the blue cells, but if not properly planned and leveraged, cardiac biomarkers can be your enemy as shown in the black cells. The positive edge of cardiac biomarkers includes the following. They can be extremely helpful in predictive population enrichment, increasing the number of events in your study and matching the right population with the right trial. As surrogates, they can serve as important efficacy endpoints for early proof of concept studies. Cardiac troponin is commonly used for safety monitoring as a marker of myocardial injury or cell death. And they can be used for a priori risk stratification, either pre-randomization or for pre-specified post hoc analysis. This is especially useful for large heart failure studies with a broad range of NT-proBNP. Now, the negative edge of cardiac biomarkers includes the following. Every biomarker has intrinsic limitations in the number available. As mentioned, we currently have two broad categories, natriuretic peptides and cardiac troponin. Also, they're limited in the intrinsically in terms of what they are intended to represent. So are the biomarkers being leveraged properly? Cardiac biomarkers also commonly lead to higher screen failure rates. Is the biomarker level essential and accurate for identifying the intended study population? We have found that there is a high degree of heterogeneity in terms of biomarker availability at sites. Not every site tests every biomarker. Moreover, different sites use different assays. So there is also much intra-lab and intra-lab assay variability. What is the methodology for validating biomarkers at your central lab or at your local labs? And then lastly, have you accounted for possible alternative biological sources of that biomarker? Is it exclusive to the process that you're measuring? For example, we've come to learn that cardiac troponin does not always equate to myocardial infarction or cell death. And a good example of this is the GALACTIC trial, in which increases in cardiac troponin did not necessarily correlate to increased levels of myocardial ischemia. Next slide. Now, to set up our next speakers of the webinar, I would like to present a hypothetical trial to illustrate some of the challenges we face when biomarkers are used in CV trials. This is a purely fictitious trial, but with realistic design and challenges, any likeness to any published trial is certainly unintentional. So this trial is named ToughRef, designed specifically for HFrEF. It is a phase 2A open label proof of concept exploratory trial, 40 subjects enrolled at 12 sites. The hypothetical IP is IM007, and the primary objective is to evaluate the effect of a 16-week treatment of IP on cardiac pharmacodynamics, including transthoracic echo assessments and cardiac biomarkers in stable chronic HFrEF. The primary endpoint is change in baseline to week 16 in LVEF and NT-proBNP. Some of the eligibility criteria are listed. So it's men and women 18 to 85 with documented HEF-REF, NT-proBNP level greater than 300 picograms per mil at screen, and all patients need to be on guideline-directed medical treatment. AFib is allowable if adequately anticoagulated. Next slide. So some of the challenges encountered in this trial are enumerated on this slide. First of all, there was a very high screen failure rate, 65%, with 2/3 of screen fails due to inadequate NT-proBNP. Moreover, only 4 sites had reliable databases. The other 8 sites, the databases proved to be not very helpful. 30% of patients enrolled were on Entresto. We know that Entresto certainly lowers NT-proBNP levels. 25% of patients enrolled actually had chronic AFib, and we know NT-proBNP levels are higher in those patients. So not surprisingly, there was a large splay of NT-proBNP levels from 300s to the 2000s. An interim analysis was performed after 50% of patients finished treatment and had evaluable data. There were inconsistent changes in NT-proBNP at week 16 compared to baseline, not surprisingly. However, there was a trend to improvement in the mean LVEF by 3.5% mean at week 16 compared to baseline. This was not statistically significant, but it does beg the question if the study design had been optimized and the eligibility optimized, perhaps a significant improvement would have been measured. This hypothetical trial was meant to underscore a few common challenges and confounders and pitfalls associated with the application of biomarkers. Kelly Milham and Liz Moore will be highlighting some mitigation strategies and a case study in their section of the webinar that addresses some of these issues. But first, I would like to invite Dr. Januzzi to discuss how we might gain efficiencies in clinical trials with biomarkers.
Yeah, thank you very much. And it's so great to join you all today. And what Dr. Lee just showed is exactly what we deal with in the execution of clinical trials and the challenges faced, particularly when we're talking about relatively small studies like phase 2 studies where biomarkers actually play a larger role in many ways because we're looking at mechanistic associations rather than hard outcomes. These challenges may be felt even more, but as is shown on the current slide, biomarkers are here to stay and they will continue to grow. And may ultimately take an even larger role with respect to safety, understanding pharmacodynamics, their roles to ensure the proper patient population. So when we say diagnostic, it's not just, hey, does this patient have a disease? Yes, no. But as we get closer and closer to a more precision-based phenotypic approach to therapeutic development, that's where biomarkers are going to play a very important role. But we have many, many things ahead of us in order to achieve that goal of linking the appropriate phenotypic identification with the therapeutic, because that's based on the prognostic response of that group. So tying together the diagnostic and prognostic nature of markers into a companion diagnostic, or theragnostic as some have said, is really the ultimate goal. And that's based on shared pathology between a biomarker and the therapeutic that we're using. Next slide, please. So when we think about the current state, this is actually kind of an interesting slide. We generated this actually a few years ago. It was reproduced in a review article just published, where we looked at the current state of biomarker use in heart failure trials. What we found was actually somewhat interesting. I think if I polled the audience and asked you what you thought markers were mostly used for, you might say, well, you know, they're commonly used as an inclusion or exclusion criterion, or maybe retrospectively as an endpoint. But it turns out actually, you know, while I would have thought that as an inclusion or exclusion was the most common indication. If you set an endpoint, you'd be correct. And that's actually very interesting because in order for a biomarker to be a valid, robust endpoint, you need to keep in mind, of course, that there need to be very careful associations between the biomarker and the therapeutic such that they're lined up on the same biological pathway. And I'll get to that in a second, because that ultimately has been the stumbling block that has prevented us from identifying companion diagnostics to speed up clinical trials. Next slide, please. Importantly, when you look at the role of biomarkers across a wide range of different applications in clinical trials, such as, for example, target engagement, monitoring the disease under therapy in order to provide a framework for understanding how a drug might retard progression or reverse disease, as a straight up prognostic marker. Kind of boring, but that's largely what we do these days with biomarkers. But as a predictive marker, that's really the holy grail, right? Which is to take a biomarker, measure it, and say, okay, this person's going to respond to the specific therapy. We haven't gotten there, but there's a path that has been clearly identified in recent years about how we might. Diagnostic, I've already mentioned, stratification, and then we get to the endpoints question. And actually, we have a prettier version on the next slide of all of these different applications. So if we can move forwards, just one, please. When we think about the endpoint role, I mean, ultimately, what we're talking about is tying together the biology of a marker together with the therapeutic intervention. And the issue is that it's not so straightforward to simply grab a biomarker off the shelf, measure it, and then claim that a therapy, if it reduces the biomarker, has anything to do with the biology that the biomarker is reflecting. And the prime example that I will give you is the now well-validated link between changes in natriuretic peptides following treatment with neprilysin inhibitors, with sacubitril valsartan. And what has been shown across numerous heart failure therapies is that treatment with, say, a beta blocker or an ACE inhibitor certainly does reduce natriuretic peptides. And when natriuretic peptides are reduced after therapy, the prognosis is better. But that does not in any way indicate that there's a direct biological link. And indeed, when we think about the reasons why surrogate endpoints fail, inevitably it often speaks to an intermediate step that we're not considering. And so the reduction of natriuretic peptides after ACE inhibitors, for example, is more likely related, in fact, to the reverse remodeling effects of the drug, which reduces wall stress and thus reduces natriuretic peptides. On the other hand, when we use neprilysin inhibitors, they cause a pharmacologic increase in vasoactive substances, including and especially atrial natriuretic peptide, which in turn directly suppresses release of the B-type natriuretic peptides. So there's an example of how a therapy is completely in line with the biological release of a marker, and in turn, changes in natriuretic peptides after sacubitril valsartan are strongly associated with cardiac remodeling, improved outcome, improvements in symptoms. That is a shining example of lining up biology with therapy. But unfortunately, numerous challenges exist on the next slide that have, and go forwards, please. Can we go back one? No, forwards. There we go, sorry. That have slowed down our process in terms of how we think about the use of biomarkers. So what do we need in order to get markers to the forefront? We can use the example of sacubitril valsartan and natriuretic peptides as a prime example. Strong knowledge of biomarker biology is going to be critical in order to link the impact of an intervention with the changes in biomarkers. So that's important because the marker really should reflect the biological effects of the drug mechanistically. In addition, very importantly, we need to establish robust sampling protocols for obtaining samples and for the measurement of biomarkers. The pre-analytical aspects in clinical trials are frequently under-recognized as being critically important, specifically and particularly for large trials, and particularly if you're measuring a biomarker that may be finicky with respect to its behavior in vitro. So, you know, if you have a temperature sensitive marker, you can't leave a sample sitting at room temperature for hours. You cannot leave it sitting even in a cycling -20-degree freezer. All of these factors are sometimes not considered by clinical trialists who do not have the background that many of you do about the role of markers in clinical trials. And then lastly, as I've already made numerous points about, the effect of the intervention should be reflected in the changes of the biomarker. And it's really striking to me how often we see biomarkers used in clinical trials with rather tenuous links to the things that are being monitored. And so I'll give you a couple of examples. I've mentioned the natriuretic peptide example in heart failure, but how about the example that Dr. Lee mentioned, the use of troponins as a toxicity marker? So why would we use toxicity markers? Well, obviously, it's an attractive idea to use a biomarker to identify the potential for heart injury, for example, if we're using a drug that may be cardiotoxic, right? The issue is that if you do not completely understand the biology of the marker that you're using, and it performs in an unexpected way, how confident are you that changes in troponin after a therapy such as, for example, a myosin activator like omecamtiv mecarbil identifies myocardial injury, which is what we have always said a rise in troponin may reflect? And indeed, in the GALACTIC study, rise in troponin was completely unrelated to cardiac remodeling. It was completely unrelated to clinical outcomes. And so what this essentially says is that the drug is doing something to troponin that has nothing to do with cardiac toxicity. And these are the kind of signals that can be well identified and elucidated well ahead of the execution of large phase 2, phase 3 clinical trials. And they really should be. We shouldn't go barreling ahead thinking that we understand everything. Another example, these are great little cocktail party examples, is the rise of natriuretic peptides as a consequence of treatment with anti-androgen drugs. Something that none of you may know, but androgens actually cause a reduction in natriuretic peptides. It's why men have lower NT-proBNP values. In clinical trials of anti-androgen therapy, sponsors previously were using markers to monitor for toxicity and noticed a rise in natriuretic peptides after initiation of anti-androgen drugs. And this led to a big concern that the drug might be cardiac toxic when in fact it was just unmasking normative physiology. So we face a lot of challenges. On the next slide, when we think about how to approach the use of markers in clinical trials, we have made a call, and the manuscript cited here actually summarizes a lot of these points. We have made a call for sponsors, for CROs, for AROs, for trialists to think very carefully about sampling strategies. There's no uniform standard for when and how to sample blood during a clinical trial. But importantly, the more uniformly we approach this issue, the more we can line studies up. So sampling strategies, how to draw the blood, how to process the samples, where to store the samples, how to store the samples are all critically important. The pre-analytical issues that affect sampling and storage cannot be understated in terms of their importance. Another error that is frequently made that leads to inefficiencies is we'll treat patients with a promising therapy and then we'll measure stuff after the study's over to figure out how the drug works. That is the wrong way to do it. And I'll make some points at the end of the talk rather, that addresses how I feel we might wish to reconfigure our thinking rather than doing this retrospective look that is often done, because this leads to a lot of ambiguity if a signal is found about the link between biomarker and pathology that cannot be elucidated from a non-mechanistic study. And what this leads to, unfortunately, is the cow path, the fact that we walk the same path frequently with clinical trials and fall into what's called the usual suspect syndrome. On the next slide, when we think about The Usual Suspects, that was a great movie in the past. This is not a photo from that movie, but the usual suspects in clinical trials in heart disease really come down to troponin and NT-proBNP. Often troponins are used for toxicity marker monitoring. NT-proBNP is often measured as a let's see if our drug is helping the patient type of outcome measure. And the problem is, as I've made the point, you can't utilize the same two biomarkers for every single application in clinical trials, in part because the biology may vary dramatically from therapy to therapy. Furthermore, it reveals a real lack of imagination and misses the opportunity for an inductive discovery approach to better understand biology of whatever disease we're treating, as well as the therapeutics that we may use. So how can we get to a more functional future state? Next slide. Well, you know, in a think tank with the FDA, NIH, and others, we had a discussion around the place where biomarkers may play a role. And among the different factors to facilitate a better future state includes a substantial improvement in our foundational knowledge. And this includes biological qualification of markers, perfection of reference range understanding, robust baseline data in diseased populations. We often identify how these markers look in non-disease, but understanding biological variation and other important analytical factors is necessary. Furthermore, it's important to recognize that one may identify a very robust biomarker, but hit into regulatory hurdles. And this is where close relationships between sponsors and regulators is critically important in order to make sure that whatever markers we utilize are qualified or have a reduced time to qualification, and then acceptance criteria from the agencies in order to make sure that we're working together hand in hand. Importantly, when we start thinking about the unique ways that biomarkers can be incorporated into a regulatory strategy, it's not so unreasonable to expect, for example, that a drug can demonstrate target engagement and a favorable change in a natriuretic peptide, troponin, or some other inductively identified biomarker in order to gain initial approval, with long-term follow-up for impact on prognosis as part of the subsequent expectation for approval. It's in some ways a modified approach for how diabetes drugs were approved, not using a biomarker. The biomarker in their case was a reduction in blood glucose, but then the longer-term follow-up of outcomes in the cardiovascular outcomes trials was pivotal to maintain the approval. So this hand-in-hand approach is going to be critically important. But importantly, we can't, again, fall into the usual suspect syndrome, and that's where we need to think about a new focus. Next slide. And this really gets to the point that I was making at the top of the table in the previous slide, which is the development of what I refer to as a biomarker phase 2, which is to say that once a therapy, a promising compound therapy, whatever it may be, has been identified, and the interest is to identify an impact on prognosis for disease X, Y, or Z, rather than treating a bunch of patients in a phase 2 trial, looking at general mechanistic endpoints and then barreling on to phase 3. Why not start earlier in phase 2 and having a specific, what I refer to as a biomarker phase 2 study, which is to apply an unbiased inductive approach using omics, proteomics, metabolomics, genomics, transcriptomics, whatever, to identify plausible drug marker pairs. So rather than just focusing again on natriuretic peptides, as well as potential druggable targets. This could actually also lead to the identification of novel pathways that might ultimately be translated into therapeutic intervention. So again, thinking earlier about identifying markers, thinking more inductively, utilizing unbiased or somewhat targeted proteomics, and then identifying markers that can be qualified for regulatory use is a very valid and unfortunately unexplored strategy at this point. But the future state could very much look like on the next slide, which is a broad-ranging combination of new or previously discovered markers, together with possibly other omics and, of course, clinical data, to identify pattern integration models that can be translated into better prediction for response, reduced risk, as well as monitoring for drug safety and response. Ultimately, this is a more functional and efficient way, in our estimation, for how markers might be able to fit in. And while I realize I've given you more of a philosophical, general approach to the question, rather than the more practical, how do we get, for example, a biomarker at every institution that can be used to enroll patients, we can talk about that in the question and answer session. This really is, you know, from a biomarker researcher perspective, the more functional future that we would hope for. So I'll stop there, and I'll pass it on to the next speaker. The goal now is to identify strategies to mitigate operational challenges. So we're going to get practical again, and I think that's going to be a really great discussion after we're done to talk about how we've been able to overcome these op challenges. So I'll pass it on to the next speaker. Thanks very much.
Thank you, Dr. Januzzi. So until this future of biomarkers arrives, clinical trials will continue to include and learn from the markers of the present. And as we know, regardless of any trial's lofty scientific aspirations, they are moot if we cannot enroll the study. So a partnership between the scientific and the operational aspects of biomarkers use are therefore essential to ensure a study can be enrolled without compromise of the targeted patient population. Next slide. This graph is an illustration of what a successful partnership looks like, burdened with a complex study design, infinite endpoints, and restrictive eligibility criteria. This phase 2 study in the HFpEF population struggled to enroll for almost 8 months. Although the biomarker eligibility criteria, which required the patient to have a minimum value of BNP at screening, intended to define and enrich the study population, in reality, it became a significant barrier to enrollment. A critical review of recruitment data, including screen failure reasons and biomarker values, provided the medical and operational teams valuable insights into how best to proceed. And although the known pitfalls of biomarkers as eligibility criteria remained, armed with many of the lessons learned that Kelly and I will discuss next, this trial was able to move forward and successfully complete enrollment with an essential protocol amendment at month 8, shown here by the hatched vertical line. This amendment, in part, provided needed flexibility within the inclusion criteria that lowered the BNP threshold requirements without compromising the integrity of the targeted study population and scientific hypothesis. And with this change, you can clearly see the rapid increase in enrollment that occurred, including both newly identified patients as well as previously screen-failed patients that were now eligible for inclusion.
Okay, so as Liz has demonstrated, biomarker selection can have a significant impact on enrollment and timeline, as well as our projections of enrollment as we're using it as an eligibility criteria. So thinking about when eligibility criteria are developed, sponsors typically will use comparisons to similar trials. They'll take into account current guidelines, but we also want to be thinking about how those guidelines may have changed since the prior, how those guidelines may have been implemented since the prior studies were initiated or completed. So considering the outcomes of those prior studies, it's always important to think about when that prior study was initiated. And at that point, as Dr. Januzzi mentioned, did that prior study then demonstrate a significant lowering effect with its target product? So as he mentioned, and if NT-proBNP were lowered with the target product, perhaps it's something to consider in a future trial, whether to exclude or take that into consideration when developing eligibility. As I mentioned, we may need to allow ourselves more time than we initially planned. So with the case study that Liz just provided an overview of, obviously the team didn't expect initially that enrollment would be so slow and had to make some changes in the moment in order to speed things up. So it can be really difficult to predict the impact of our eligibility criteria, especially as it relates to biomarkers. But once we have the eligibility criteria, it's really difficult to change the protocol once it's underway. So during that time, when we're not able to change the protocol, we're not able to do anything differently in terms of our eligibility criteria, what can we do in order to help support enrollment and help support sites in enrolling patients into the trial, despite some of the challenges with the criteria that we've been prescribed? So first up, it's really important to ensure a smooth experience for the patient. So we're not wanting to expose study subjects to unnecessary procedures or increased burden at all. So one way that we try and mitigate that risk is to tier our screening procedures in order to reduce invasive procedures and minimize costs where we can. So thinking about in our screening activities, if possible, to have the laboratory procedures completed first, and then await results before moving the patient through to any imaging or physical function tests that may be required. Especially in a at-risk population, as many as of our heart failure or advanced cardiovascular studies are, we want to avoid stressing the patient unnecessarily if we know that, for example, their blood work comes back and they're not eligible. So really considering that when the patients are in the trial will help us to ensure that patients have the best experience that they can. Another way that we mitigate some of the challenges in encountering biomarkers at the screening visit is using pre-screen testing or historical values as a guide to help investigators decide who is the best person to bring in for the actual screening visit and all of the remaining screening activities. So we have implemented in some of our studies a pre-screen consent form so that testing can be done locally, or we allow sites to use historical values to provide some of the flexibility without risking the patient population that we're under study and without risking allowing patients in who maybe will dilute our patient population that we're looking at. As Dr. Januzzi also mentioned, sensitive biomarkers can be affected by temperature, but they can also be affected by time of day of collection, of fasting conditions, and we want to make sure that sites are fully trained and thoroughly understand the risks to those laboratory samples. So often when the scientific team is developing a protocol, they implement a biomarker, but then it's up to the trial management team and the laboratory team to really make sure that the sites are trained and understand the risks of collecting sampling at the appropriate time and under the appropriate conditions, and also storing them and shipping them under the appropriate conditions so that we get the correct data out of the assay. So making sure that we're providing guidance to sites also helps to make sure that we've got the most reliable results that we can get. And then finally, it's really critical that we're discussing biomarker impact with potential clinical sites during the feasibility process to make sure that we're selecting the right site. So we can go to the next slide. Once we have those sites selected, obviously we're going to be targeting sites that utilize biomarker testing in their standard of care practice if we can. So if sites do have historical data available, that's wonderful. That really helps us to understand our projections, our impact on how the biomarker might affect us. But as we all know, sometimes biomarkers can be variable and sometimes the historical value doesn't always predict future. But what we do know is that sites that consistently use this biomarker in their standard of care practice are more experienced and better able to predict which patients might be eligible based on their historical information. So for those sites that don't use biomarker testing in their standard of care practice, what we do is also help support them with the database mining efforts. So perhaps they don't have NT-proBNP or high sensitivity cardiac troponin results in their database, but what they may have is a standard BNP, or they may have other laboratory values that we can help them to use to deduce whether patients would be eligible for the study. So one way that we do that is help sites to develop their search criteria, whether that be through an EPIC, SLICE, or DICE, or other search criteria. We often work with IT teams at clinical sites to help narrow the searches and make them more realistic. Often we hear from sites that when they do their initial database search, they come up with hundreds, if not thousands of patients, and it really does take a lot of effort to narrow that down. So as much as we can help sites to successfully mine the database and narrow down the patients. It saves time on the site's behalf, but also we're not calling patients unnecessarily in for screening who ultimately are not eligible because we don't have enough data in the history. And then finally, making sure that the sites are truly educated on identifying the most appropriate patients for the trial using the biomarker is also important. So as we are expanding and we're seeing that cardiac biomarkers are being used in other therapeutic areas, it's really important that we train the PIs for our trial on how the biomarkers are expected to behave. So for example, a study in nephrology may include cardiovascular endpoints to demonstrate safety of the drug. So in that scenario, we would develop materials for the investigators to explain what the biomarker does, how it acts, what it acts on, what is the biophysiological pathway that the biomarker is working on. How might patients with this condition be affected by that? And then also, how is this biomarker expected to change as a result of the study? So really making sure the investigators are fully educated on our endpoints and not just saying, oh, you know, this is just another lab that's going to be in your blood sample collection. You know, draw your chemistry, your hematology tube and send it in. Really making sure that the investigators are on board with our endpoint plan, with our study plan, to make sure that they are also properly selecting patients so that the study is successful. Okay, and we can hand it back to Liz.
And finally, some points to consider when utilizing biomarkers in trials for monitoring and assessing safety. During protocol design, several key logistical questions need to be addressed, starting with what markers to include in the study. The careful selection of biomarkers and assays that will be feasible at targeted sites is essential. Site access to specific assays will vary widely or may not be available at all. This may limit the use of local labs for those pre-screening and screening activities that Kelly just spoke about at the site, resulting in lost efficiencies and increased cost if central labs must be used. In support of safety surveillance, choosing the right biomarker for the job can be equally challenging. Although we utilize biomarkers such as BMP and troponin within the clinical environment as standard practice, their application within clinical research safety activities can be less clear, particularly when the effect of the investigational product on the biomarker and population is unknown. Therefore, to complement biomarkers, the inclusion of additional safety procedures such as imaging, vital signs, safety labs, and ECGs will support safety and risk mitigation needs. Additionally, although the use of central labs to minimize variability across sites is preferred for endpoint analysis, for safety activities, splitting up the safety labs, such as troponin, allows the site to utilize local labs to ensure a more rapid result and response to any urgent safety issues. Again, interpretation of selected biomarkers is challenging. As Dr. Januzzi discussed earlier, a solid understanding of biomarker biology and temporal relationships between the biomarker abnormality and event is needed. Multiple confounding variables may also be present. For example, the use of BMP should take into account potential differences within specific phenotypes and characteristics of patients, such as obese HFpEFers, where circulating levels of BMP may be decreased. Acknowledgement of such variables such as race, renal function, BMI, and gender within the protocol and safety monitoring thresholds minimizes the impact to enrollment, as well as within safety and risk evaluations. And as we continue to build upon our knowledge and application of biomarkers within clinical research, what is clear is that in collaboration with other routine safety activities, such as aggregate trend reviews and clinical data surveillance, biomarkers do support the overall safety and risk management of patients. Conscientious selection of markers, clear safety parameters, follow-up guidance, and stopping rules further promote the safety and protection of the vulnerable populations under our care. Because again, at the end of the day, it should be all about the patient. Next slide, please.
Thanks, Liz. So Kelly and I are going going to summarize the webinar, our main points in this slide, and I do think we will have time for a handful of questions to answer. What we've talked about today will hopefully will help our audience be informed on how to sharpen the edge of CV biomarkers. In drug development, it is critical to balance those pros and the cons of application of cardiac biomarkers and to use them carefully. We hope that our discussion with Dr. Januzzi has highlighted those challenges, and Kelly and Liz have provided some mitigation strategies in terms of clinical trial management. So I think important questions: when do biomarkers add value? When are they a detriment? And as Dr. Januzzi has said, how are they linked to the intervention being measured?
Yeah, I think it's important to understand how our biomarkers and what we currently have available are impacting the studies that we're designing.
For drug development, as Dr. Januzzi says, we need to understand biomarker biology. Adaptation of the use of biomarkers during a trial is also quite important. Being able to avoid the common pitfalls is integral to trial success. And hopefully the case study that Liz presented highlighted some of those issues, and the presentation that Kelly presented about the importance of pre-screening. And lastly, our field is in need of the development of better biomarkers so that we can go beyond the so-called usual suspect. I do think we have some time for questions, so I'll hand it over to Ryan.
Thank you, all of you, for your very insightful presentation. I'd like to invite the audience to continue sending their questions or comments right now using the questions window for this Q&A portion of the webinar. We've already received some of our questions today, so we'll get ourselves started with those. Our very first question is directed to Dr. Januzzi, and it asks, what's your take for using EKGs as a biomarker to monitor cardiac safety?
Yeah, it's a really important question. You know, there's a sort of long tradition of using EKGs for monitoring safety, but the simple truth is that while EKG monitoring provides information about, for example, prolongation of repolarization, which might increase the risk for torsade, a long time-tested and time-honored role for ECG monitoring in clinical trials, the information gained from the electrocardiogram is relatively modest. And so, you know, it falls into the same category as some of the biomarkers I showed that are the usual suspects, right? We go to EKGs because that's what we've always done. But the simple truth is that they don't provide as much information as we might like about things that their EKG is not really designed to look at. So, you know, yes, coronary ischemia might be detected, but EKGs are a snapshot in time. The patient may or may not have evidence for ischemia when it's taken. You know, arrhythmia also can be a here and gone kind of thing. So it just speaks to the need for a more comprehensive strategy for safety monitoring. And that's why we have turned to biomarkers. And to be sure, despite the challenges that we've talked about, there's definite upsides to using markers like troponins for risk monitoring. But still more information is needed in some cases.
Excellent. Thank you very much for that answer. The next question is also directed toward yourself. It would like to know, what are the new biomarkers that move us beyond Framingham score?
Yeah, that's a really interesting question. So the Framingham risk score, for example, for predicting ASCVD, again, time-tested, time-honored, but clearly from a discrimination perspective, leaves something to be desired. And can we identify better strategies for identifying patients at risk for arteriosclerotic cardiovascular events? And the answer is definitely yes. And this gets back to the whole idea of using an inductive approach with omics to develop a better strategy for this application. For example, we published a paper in 2016 looking at a proteomics strategy with leveraged by artificial intelligence to develop a 4 biomarker panel from which an estimated likelihood for the presence of obstructive coronary artery disease was generated. And with this, we had an area under the receiver operator characteristic curve of nearly 0.90. It was 0.87. And that's the kind of discrimination that we're talking about when we're looking for a really high fidelity signal. That's about as good as natriuretic peptides for heart failure diagnosis, and well better than the Framingham score to identify obstructive CAD. So ultimately, you know, with innovation and a focus on moving the needle, no pun intended, forward from the places that we have been, I really do feel confident we will be able to develop that kind of useful strategy to, for example, trial out therapies to reduce arteriosclerotic cardiovascular disease progression and adverse outcome.
Excellent, fantastic, thank you. We have a question here for Kelly. The question would like to know, how have you approached pre-screening for biomarkers when sites may not have availability of that specific test, for example, HSTNI? Further, do you automatically exclude them as a potential site then?
Sure, thanks for the question. So in general, if sites do not have availability to perform a test locally, we will explore options for running pre-screening labs through a central laboratory like Medpace reference laboratories. We have certainly done that in the past for sites who are unable to collect samples locally. So no, we wouldn't automatically exclude them as a potential site if they've not got historical values available. Rather, we would make sure that the site has the appropriate patient population, looking at other data that we have available, patient medical history, patient diagnosis codes, that type of thing. And then for screening, or if we need to pre-screening, we can manage samples through a central laboratory.
Fantastic, thank you. The next question is back to you, Dr. Januzzi. It would like to know, are there other biomarkers that have promise for application in heart failure?
Yeah, thank you very much. So I'll start the answer by first adding that there has been an increased interest in managing the situation that was just mentioned, which is the inability of a site to run a specific marker because they don't have the availability on site. So for example, finger stick testing for natriuretic peptides, a finger stick NT-proBNP assay, that's something that a number of different companies are looking at. In addition, point of care strategies for high sensitivity troponin are coming as well. So that hopefully should mitigate some things, but it's really critically important just to emphasize, if that kind of a strategy is taken, you have to be very careful about mixing assays. So, if one site runs high sensitivity troponin I and another site runs conventional troponin I, that's a no-no. And furthermore, if they run another assay for high sensitivity troponin I, that's also a no-no. So there are factors that Medpace and others need to keep in mind as they deploy biomarker-based strategies for inclusion. Now, to more directly answer the question you asked, yes, indeed, there are definitely other markers that have promise in heart failure. I talk about the big three are NT-proBNP, high sensitivity troponin. We recently showed that above and beyond its role as a biomarker that's used for myocardial injury in patients with chest pain, we recently showed that troponin in patients with chronic heart failure is a very good predictor of cardiac remodeling and response to therapies for heart failure. So that's interesting and really opens up a potential application there. And then the third of the three is a marker called ST2, which is a marker that is secreted by epithelial cells in response to stretch and stress, and is also expressed by cardiomyocytes for the same reasons, and is a very good marker for prognosis and cardiac remodeling in heart failure. Now, of course, there are many, many other markers that look good, but these are the ones that have stood the test of time and validation. Ultimately, there may be others coming, and again, I re-emphasize one more time, the strategy of a combinatorial approach to marker use based on an omics measurement and artificial intelligence analysis is really ultimately what I sort of foresee in the future.
Dr. Januzzi, it's Richard. Thank you for that answer. Just to follow up on that, there are a number of approaches looking at radiographic biomarkers, if you will, looking at residual inflammatory risk for coronary disease, like FAI, perivascular inflammation. I'm wondering if you had any insights or any thoughts about using so-called radiographic biomarkers.
Yeah, thanks very much for that question. And to be sure, as defined, a biomarker is not just what circulates in the blood, right? So imaging biomarkers are, in many cases, as if not more robust, because we have direct visualization of their presence in, for example, the heart, in tissues. So late gadolinium enhancement as a sort of a general sense of a biomarker. So yes, indeed, all of those are certainly promising, but come, of course, with the challenges that are involved with imaging, the need for specialized interpretation, the need for scanners, the cost, and then ultimately whether these markers translate to a mechanistic implication, much like I said for circulating markers, right? You know, you need to make sure that the biology is completely aligned rather than the possibility of something being in the intermediate step between a marker and an outcome, which has always been a challenge with respect to surrogate endpoints.
Excellent. Thank you very much for that great question, and for all of the answers from everyone today on our panel. Unfortunately, we have reached the end of our time here today for the Q&A portion. Now, if we couldn't attend to your questions, though, the team at Medpace may follow up with you, or if you have further questions, you can direct them to the email address that's up on your screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it will help us to improve our webinars. Now, I'm about to send you a link in the chat box, and with this link, you'll be able to view the recording of this event on this page, and you can also share this link with your colleagues when they register for the recording here as well. So I encourage you to do that. Now, please join me once more in thanking our speakers for their time here today, and I hope that you all found the webinar informative. Have a great day, everyone, and thanks for coming.